Hello and welcome everyone to the Lexogen and BlueBee webinar. I'm Ekaterina Alexeyeva, Senior Marketing Manager at Lexogen, and I will be moderating this webinar today. The title of our webinar is Lexogen CORALL Total RNA-Seq Complete Solution for Whole Transcriptome Analysis. This webinar will take about 45 minutes, including 30 minutes of presentations and 15 minutes for Q&A session. Let me introduce the speakers and the topics for today. The first talk will be by Lexogen Product Manager Martina Sauer. She will give an overview of the Lexogen's CORALL Total RNA-Seq Library Prep Kit. In the second talk, BlueBee's field application scientist Andreas Klingenhoff will present the BlueBee Genomics Platform and an overview of the CORALL pipeline on it. Afterwards, we will have a Q&A session, and we invite you to submit your questions by typing them in the question box, which you can find on the right panel of the webinar software under the quote sign with the question mark in it. Feel free to type in your questions during or after the talks. However, please be aware we will only answer them after the talks during the actual Q&A session. We will try to answer as many questions as possible as long as the time permits. And now it's my pleasure to turn it over to Martina for the first short talk. Martina, welcome, and please go ahead.
So thank you, Ekaterina, for the kind introduction, and also a very warm thank you to BlueBee and all the organizers on the BlueBee side for this webinar. It is my big pleasure to introduce you all to Lexogen's new CORALL Total RNA-Seq Library Prep Kit, to its features, and also to give you an idea of the data analysis pipeline that our scientists have developed for CORALL, and that is now available on BlueBee, as Andreas will later on describe in more detail. Before we begin, I want to give you a brief introduction about Lexogen, who we are, what we do. So Lexogen is committed to develop and to deliver end-to-end solutions for RNA sequencing. We are founded and our headquarters is based in Vienna, Austria. We are a very multinational team, and we also have a subsidiary in the US, in New Hampshire, where we also have sales representatives and tech support, as well as we have them here in Vienna. Also, we have a worldwide distributor network, which you can also find on our website. So as Ekaterina has already introduced, today's talk will be about CORALL. As I mentioned earlier, Lexogen is committed to providing end-to-end solutions for RNA sequencing. I have here depicted the RNA sequencing pipeline, which starts from sampling of your material to sample preparation, sequencing, and finally the data analysis. Today, we also only focus on two of those parts that are covered in this talk, the sample preparation and the data analysis. Today, we will talk about the whole transcriptome sample preparation kit CORALL and the corresponding data analysis pipeline, which is implemented in BlueBee, and Andreas Klingenhoff will guide you through that later on a bit more. So now, I want to jump straight into CORALL. So as I said, CORALL is a whole transcriptome RNA sequencing library prep kit. And CORALL delivers complete transcript coverage from start to end. It is an extremely fast and easy library prep protocol, and all components that you require for the library preparation are included in the kit. CORALL is also suitable for low input and for FFPE samples. We have benchmarked CORALL's performance to most commonly used library prep kits currently available on the market. And our focus of today's talk will be the data analysis pipeline that is available on BlueBee. So Before going into some data about CORALL and its performance, I want to give you an introduction about how the chemistry of the CORALL library preparation works. So first of all, you have to know that CORALL is an RNA fragmentation-free protocol. So in contrast to most other library prep kits on the market, we do not start by fragmenting the RNA, but we leave them as intact as it can be. This minimizes the bias by physical RNA fragmentation. The insert size in your final libraries is defined by the distance between the random primers that we use. We use the so-called strand displacement stop primers that randomly hybridize to your RNA template, and the reverse transcriptase will perform the reverse transcription until this first strand synthesis hits the next displacement stop primer, and this will define the length of the final insert of your libraries. In a second step, we ligate the linker oligo that already contains the UMI, the unique molecular identifiers, very efficiently to the 3' end of the first strand cDNA. And via this mechanism, you result in these short fragments that contain the UMI and the inserts. This way also maintains the strandedness of your RNA. And all these Lexogen proprietary steps together result in a complete transcript coverage. On the next slide, I want to show you how this complete transcript coverage looks like on an endogenous gene. Here I show you an example of GAPDH, so a very well-known housekeeping gene. And as you can appreciate here, that compared to commonly used kits currently on the market, we have also very nice, very complete transcript coverage over the whole entrance structure. What you can also appreciate when you zoom in to the very 3' end here, you can see that the reads generated by the CORALL library prep kit cover the whole transcript to the very 5' end of the transcript, whereas other methods tend to not really reach those last couple of nucleotides. Also, you can appreciate on the other end, the 3' end, this transcriptional stop site, that also here the coverage of CORALL is more complete. Also here on this slide, you can see that it's a difference of a whole transcriptome library prep kit to merely quantitative methods such as Lexogen's QuantSeq 3' mRNA-Seq or microRNAs, where you just obtain quantitative information of your data. With CORALL, you obtain the quantitative and also qualitative data from your RNA samples, which allows you also to assess isoform discovery or isoform quantification, but also it would allow you to, for example, re-annotate transcriptional start and transcriptional end sites. And you can also use a CORALL for for de novo assembly of transcripts. On the next slide, I want to point out that we do not do any cherry picking here when I show you an endogenous gene. We have done this analysis of the 5' and 3' ends on the ERCC controls. So ERCCs are spike-in controls that have known transcriptional starts and end sites. And when we align all the known transcriptional start sites of the ERCCs and align all ERCC reads to this very 5' end, you can really see that they accumulate on a nucleotide resolution accurately to the very known start site of the ERCC. And the same is true for the 3' end, where the coverage is more intense than for other kits currently available on the market. Another feature of CORALL that is very much appreciated by our customers is that it's a fast and easy and all-in-one library prep protocol. So it only takes you 4.5 hours to generate libraries from your starting material. The protocol is endorsed for its very easy workflow, even if you have not too much experience in the lab. If you follow our detailed user guide, you are able to generate reproducible libraries. And also the components are all included, and you do not need any additional time to incorporate some add-ons, for example, such as dual indexing. The input requirements for CORALL are extremely flexible. You can go down to 1 nanogram of input total RNA as input into the ribosomal RNA depletion or mRNA enrichment with our KIDS RiboCop or polyase selection. If you already have pre-enriched RNA, you can even go down to 100 picogram of input. Also, our CORALL protocol is compatible with low-quality samples such as FFPE, as I will point out later. CORALL is also highly cost-efficient, so all the required components included. For example, we have included the UMIs, the unique molecular identifiers, the I7 indices that you usually have to buy separately from other suppliers. Also, the purification beads are already included. The whole protocol is very automation friendly. So if you want to automate CORALL, you can use the kit as we send it to you. It is very easy to automate with liquid handlers. You do not need any additional kits for automation. And we here at Lexogen have also very close collaboration with the manufacturers of liquid handlers and can help you out with providing protocols for those machines. And last but not least, data analysis is available on the BlueBee Genomics Platform, as we will talk about later. So I mentioned that the CORALL library prep is suitable for low input and for FFPE samples. So let's start with the low input material. As you can see here on this gene discovery plot, that we have performed on different input material from 1 nanogram to 10 and 100 nanogram input into RNA depletion and subsequent CORALL library prep. And you can see that gene detection rate is almost as efficient when you use 1 nanogram input as when you use 10 nanogram or 100 nanogram input. So it's very robust and very reproducible among the input scale. The very same is true for using FFPE samples, so formally paraffin embedded samples. Here you can also appreciate an almost equal gene detection rate from FFPE and fresh frozen material. And if we further analyze that data, you can also see that the overlap of genes detected from both kinds of material is very large, about 90%. So you can use FFPE material almost as good as fresh material. And last but not least, you want to know how CORALL performs in comparison to other kits on the market. So of course we have benchmarked CORALL's performance to the most commonly used kits that are available. And on this gene discovery plot here again, you can appreciate that CORALL has an efficient gene discovery rate as currently most frequently used kits on the market. And also in terms of the actual data that comes out, you can see here that 96% of the commonly detected genes overlap between CORALL and each of the analyzed competitors. So this very nicely illustrates that you can interchange CORALL library prep with other whole transcriptome RNA-Seq library preps currently available. Now, one more point that is a bit different between CORALL and other kits on the market. We also provide you with a data analysis pipeline, because we know that data analysis is actually in the end the major part of your whole experiment, and we want to support our customers. And we have invested a lot of research and development into our data analysis pipeline. So the pipeline that is now implemented in BlueBee and available for you performs for you the read quality control, the mapping, the UMI deduplication, and most importantly, it provides you with a reliable transcript quantification. For this transcript quantification, we use MixSquare. MixSquare is Lexogen's proprietary software that is now embedded in this pipeline. MixSquare does precise transcript abundance estimation, thus will lead to an accurate detection of differential gene expression in downstream DE-Seq experiments, for example. MixSquare provides an exceptional reproducibility across variable sample conditions, and also very important, it has fast run times and a small memory footprint. So in the next couple of slides, I just want to give a very rough introduction into MixSquare, why we choose it and what it does. So in terms of QuantSeq transcript abundance estimation, most algorithms that are available assume a very equal coverage across the gene, but that is usually not the case. Sometimes you have a bias, in this case, for example, on the 5' end, and most methods to estimate transcript abundance cannot really deal with these kind of biases. What MixSquare does, it mixes a mixture model to the data, and that results in that MixSquare can learn isoform-specific bias from the reads that you have seen in your RNA-Seq experiment. If you want to know more about MixSquare itself, you're welcome to visit our website. There's a lot of data on MixSquare, how it performs and how it has been developed. MixSquare is the published pipeline. We have published it a couple of years ago, so you can go into depth there. So now we come to the reason why we use MixSquare. Many customers want to do differential expression analysis after they have estimated the transcript and isoform abundances. And here I show you an experiment of RNA isolated from blood from 2 different donors, and we have performed a library prep with RiboCop, our RNA depletion, and subsequent CORALL library prep. We have done this in various numbers of technical replicates. And after using either MixSquare on this side or string type for transcript abundance estimation, we have performed a differential expression analysis using DE-Seq and analyzed those results on a PCA plot, a principal component analysis. So in this case, the principal components that is responsible for the largest variance is the biological source. In this case, it's the donor. And as you can see here, when we do this PCA analysis on string type data, you cannot really see a particular clustering. The only clustering that you might be able to see is more into the replicates that we have been doing than specifically to the donor. On the other hand, when we use MixSquare for transcript estimations and subsequent DE-Seq for the differential expression analysis, the 2 different donors very nicely cluster on this principal component analysis. So this is why we have decided to use MixSquare on the pipeline, which is implemented on BlueBee, to give you really the best outcome. So this just sums it up. MixSquare for complete transcript coverage, fast benchmark performance, and suitable for FFPE samples. And for the data analysis on BlueBee, I will now hand over to Andreas Klingenhoff, who will tell you more about the BlueBee pipeline and what the pipeline does for CORALL.
Yeah, thank you, Katharina. Thank you, Martina, for the kind introduction. In this second part, I will give you a short overview of what BlueBee can do to analyze the data that you created with the CORALL kits from Lexogen. We will have a short look on BlueBee architecture, how we do things, how we deploy our software, how we can guarantee privacy compliance. And at the end, we'll look in more technical details, how the data can be analyzed, how you can register on the platform, and what results you actually get at the end. BlueBee mission, we position ourselves as a partner for companies like Lexogen to provide an ecosystem, an IT infrastructure environment where companies, partners can deploy their omics technologies, making them accessible to their customers in a robust, scalable way, that they don't have to worry about there's more technology in the background. That's the part BlueBee takes care of. As already mentioned, so a direct B2B business where we provide the IT infrastructure to our partners with, on top of the IT, a simple to use user interface that makes it for you as an end user easy to work and to analyze your data. Our partners own the IP, the solutions, so it is software IP technology, in this case developed by Lexogen. So you don't have, or you can be sure, you can rely on that the pipelines we use, that you use to analyze your data, really reflects the IP developed, the science developed by the partner you actually work with, the kits you use. And another important point, the data that you upload to BlueBee platform stays your data. BlueBee never claims any rights on the data. We will never access your data or make use of it. You have the option, of course, to share your data with Lexogen for joint research projects, for support questions. You can, of course, also give us access to the data if there might be some technical issues and we need to look into it, but you stay under control of the data. It stays your data. It's always an opt-in solution. You have to give us, you have to give Lexogen access to the data. The setup with Lexogen, slightly different view on it actually again. So we serve Lexogen to enable solutions like CORALL. It's the third pipeline that we bring to the market together. We have already there the QuantSeq and the Slamdunk pipelines. It is a ready-to-go infrastructure for Lexogen to bring their pipelines to you, to your end user, and it's also for you some ready-to-use environment. You just register, provide your credentials, and then you are ready to go. You don't have to think about investing in IT resources. You can be sure that the data is saved, stored, compliant, all the things you don't want to take care of if you do analysis like this. How does it look like? A little bit more in detail. So you are here as an end user. You do the experiments, the sample preparation, the sequencing. The data FastQ files gets uploaded to the BlueBee platform. BlueFlow, in this case, it's the generic platform where we, for now, host the Lexogen pipelines. Data is processed there from FastQ to here it's VCF. It actually should be BEM files in this case as intermediate files, and then some reports get generated out of it. Can be just transcript counts, gene counts, some QC reporting, or even some medical report at the end. End users, you can think of different scenarios of end users. Can be for clinical use, just wanting to have final reporting for decision-driven further actions, or it can be more research-focused, having access to all the intermediate files and doing some local processing research based on that data. That's the situation that we have for now. No, that's it, wrong. There is a different layer that we support, which we call BlueVantage, which is some kind of graphical interface customized for specific applications, simplifying the usage of the pipelines, just focusing on what really needs to be done, uploading data, starting analysis, and receiving some results, reports, taking away, hiding all the technical possibilities, configuration that you actually have in this layer, and that in many cases you don't need if you're just going for clinical reporting. We don't have this yet for Lexogen, but we are working on it. So there will be a BlueVantage solution that integrates the 3 different Lexogen products that we have already, QuantSeq, SlamDunk, and the new CORALL one. It is in development. It will be available early 2020, so early next year. There is another component that might be of interest, call it BlueBase. It's some data warehouse database where you can integrate data from a large number of data sets that you have analyzed, can be really results, but can also be a lot of QC data, mapping rates that you want to analyze, mine, to get a better overview if you're working high throughput, how how performance develops over time, over experiment. It's not part of the Lexogen solution yet, but might come to at a later point. If we look in the architecture a little bit more in detail, so we have here on the left side, you actually as an end user, local file system, that's where your data is. And then you interact, so if you log in, create an account, you interact with the BlueBee central platform, which is hosting the account information, all the definitions of pipelines, data transfer definitions, so how you upload data, ticketing, so all the technical account focused information. It does not contain any of the data that you upload. It does not contain any of the results that you create, no metadata. Those sensitive data, oh, it's not sensitive, but those application data stays in one of our regional data centers. So this one is actually located somewhere in Europe, but the data centers where your data is, where your data gets analyzed, also the compute happens here, can be globally everywhere. So it's defined where it is, you know where your data stays. For Lexogen, it's also a data center in the EU. But for compliance reasons, we can also define that your data goes somewhere or stays somewhere in the US, in the UK, and many other locations. Since this month, we also support data residency in mainland China. And again, data is uploaded directly to these regional data centers. The interaction actually is between you and the central platform via browser. Upload can be done via browser or can be uploaded via connector directly to the regional data center. If we extend the view a little bit globally, so it's actually the same situation again. Now you are here as an end user. We have the central platform here, and then there are different data centers globally distributed, and depending on the application, depending on the project, your data can go to different regional data centers. Compliant data residency is guaranteed, so you know that your data stays in a specific data center, you know in which data center it is, and compute is also done here. So we have regional hardware to analyze the data in those data centers, and only the results are delivered back directly to you, again bypassing the BlueBee central platform. That brings us to an overview, data centers worldwide. This is actually only a subset of what we support. We have more than 50 regional data centers we currently have access to. It's not really worldwide. It has, of course, a strong focus on Northern America, Europe, and the Southern Asia Pacific region. That's where most of the data is created at the moment. We can work with other data centers too. Again, mainland China is actually not on the map yet. We have a data center now close to Beijing where we can really host and analyze data in mainland China. We are GDPR compliant, we are ISO compliant, we are HIPAA compliant, we are HDS certified. A few more of those certificates for different regions in Europe, in Northern America, Canada, for example. So that's where we put actually quite some effort to support this data residency and compliance worldwide. That brings us now to CORALL, the new pipeline on BlueBee platform. Give you a short introduction on how to register on the platform, on how to create, manage projects, uploading data, start a run, and what results you actually get at the end. Access to the platform. You will get an activation code from Lexogen. That gives you access to the platform, and it allows you a specific number of runs defined in the activation code. For registration, you just go to the registration form on our platform, enter the code here, provide your user information, define a password, and actually then you are set already. It's done in a minute or two. You enter or you create your own specific account with a predefined CORALL project inside, which already contains the pipeline. I'll show you a little bit more details on the next slides. If you already have an account, for example, for QuantSeq Slamdunk, you can easily add your activation code to those accounts and then manage your data in one account altogether from the different applications. But it's not mandatory. Even if you have an account already, you can, of course, also register a new one and keep the CORALL data somewhere here separate. You're not limited to this one project. That's just there to make it easy for you so that you can start directly. You have, of course, the opportunity to define as many additional projects as you need to manage your data in your account if you want to give different samples to different projects. That's, of course, possible. Inside the project, what do we see? We have the settings, the general information about the project owner, data residency in your region. You can define if you want to share data between different accounts. You have the possibility to give permissions to different users in this account, to add additional users to this account. And there is a short, in the default project, a short summary what CORALL actually does, how the workflow works. Data upload to the platform. Data upload always goes into a specified project. You have different options to upload data. Most easy one is just browser upload, drag and drop, go into the data section of your project, drop your files here and they get uploaded. You can also install a connector, as already mentioned. That's a small Java application installed on your computer, establishing a defined data connection between your server and our cloud. Advantage of a connector is if the upload gets interrupted, especially for larger files, it can resume the upload afterwards, and you don't have to start from the beginning again. Third option to get data into your account, into your project, we support cloud connectors, so you can directly connect to Amazon, to Illumina's base space, or to the Google Cloud, and transfer your data from there to the BlueBee project without having it to download first and re-upload it. So that are the different ways to get data into your account. Pipeline, you have seen this image already before. It's a pre-configured workflow. Our settings have been optimized by Lexogen, so it's exactly tailored to the CORALL kit. Steps, parameters, or sets that it gives you the optimal results you want to have. At the moment, we support a little bit more than 40 different target species. Human, mouse, rat, the most important ones probably. Few model organisms like Arabidopsis, Drosophila, zebrafish, few plants, few insects, some very exotic ones. If nevertheless your species of interest, your target genome is still missing, reach out to us, reach out to Lexogen, and if the data is freely available, it can be added to the list kind of straightforward. The different steps in the pipeline. So as already mentioned by Martina, it starts with UMI sequence removal from the actual read. Then we have some adapter removal, read trimming, removing low quality nucleotides at the 5' 3' end of the read, using catadapt for that. We do some fast QC reports for the input files and for the trimmed files. So you can see the quality differences, or actually the quality increase, hopefully, after trimming. Then the next step is the star aligner, using the defined target genome. Outcome BAM file, it is available for you to download, so it's one of the intermediate files you can use. Next step is the deduplication step using the UMI tags. You also get a deduplicated BAM file, also available for download. And then the actual quantification step comes with mixed square, which in the end gives you gene abundance, transcript abundance files, and a multi-QC report summarizing all the QC metrics that have been created throughout the analysis. And as already described by Martina, we are currently using Lexogen's mixed square for the transcript quantification. How to start a run. So again, inside your project, you go to pipelines, select a pipeline. At the moment, there's only one version, version 1. There might be additional versions at a later time. So if we need to update the pipeline because there are newer versions of some of the tools available, whatever, we will release a new version. Maybe a few words on version management on BlueBee. If we update a pipeline, it will always be a new version. You will be informed about this, that there is a new version available, and the old versions will stay there. They will still be available for you. So if you need to continue your analysis and still want to use the old one because you started with that, it will still be available. Running the pipeline, just hit the start run button. Enter input files. Also, as already mentioned, we support single read and paired end. So you either provide one file or you provide read 1 and read 2. You have to provide a sample ID and select the target species, and then you can just start the run. There's no need to configure anything. You don't need to provide any parameters. That's all preset for the CORALL kits. You can, a little bit too fast. You can automate the analysis to some extent on the connector part. So if you have a connector installed for upload, you can define some execution rules, so uploaded files get analyzed automatically. If you look at the run itself, you get a graphical overview of the pipeline, input files, output files, and the actual CORALL pipeline and some pre-processing step. It's now all green here, so it means the pipeline has been executed successfully. While the pipeline is running, you can see the progress here in this graphical visualization. On the right, you have some technical details about the settings, about the input files, about the output files at the end. You can just click through that. Output files, reports that are generated after the run, you get an overview of the output files here. as already mentioned, it's the BAM files, it's FastQC files, it's the gene transcript abundance files, and it is a multi-QC report that's generated by the pipeline, summarizing all the statistics, all the metrics that have been generated throughout processing the data. All the files can be viewed online or can be downloaded directly to your local infrastructure. And with that, we are actually at the end already. I thank you for your attention, and we are open for questions now.
Thank you, Andreas. Thank you, Martina, for these great talks. We are running a bit over time, but we will definitely have time to answer a couple of questions from our audience. Before we get started with our Q&A session, I would like to remind how to submit the questions. You can type them into the questions box, which is located on the right side of the webinar platform. And I see we have already some questions. All right, our first question is that data upload for BlueBee works using a pre-issued code. What happens if the connection is lost before the upload is complete? Is the code lost, or can the upload be repeated?
The data upload actually does not require any of your credits. So data upload, data download is completely free. The credits are only used for analyzing the data, for starting a run. And if the run fails for whatever reason, the token is refunded, so you can use it again.
All right, we have another question. I saw somewhere that the BlueBee platform charges a specific price per sample. Can you tell me a little bit about how much that would be, and if it is dependent on the kind of analysis you want to perform? As an academic scientist, I want to use the best technology, but I'm sadly limited by the limited grant support.
Okay, so thank you for submitting that question. I think I can answer that one. So for the introductory period, the data analysis pipeline for CORALL is included when you purchase the CORALL kit. So we have a promotion until the end of March 2020 that with every CORALL kit, you also receive the corresponding amount of ones for free on the BlueBee platform to use within the next year after having purchased the kit. And after that, the ones on the BlueBee platform are subject to pricing, but you can contact us in case you have any problems and need to repeat some experiments. We will find a way, and I think with the pipeline with Mixsquare, you will have the best available technology.
All right, the next question, did I understand correctly that your libraries are stranded?
Short answer, yes, absolutely. So above 99% strandedness due to our strand displacement stop and linker ligation technology.
And can you upload custom genomes for alignment?
That's not possible at the moment. What we can do is to make it available to you. If you have a custom genome sequence, it's not as straightforward, not as easy as just making it available for everyone if you want to have access just for you, but it's possible, we can do.
Okay. The next question, how long does the CORALL data analysis on the BlueBee platform take?
For a regular data set, that is below 1 hour, between 30 minutes and 60 minutes.
Mm-hmm. Dear Martina, how many replicates are needed for reliable analysis? Also, did you comparison GC, VC, AT-rich genomes? How well CORALL performs in case of GC-rich genomes?
So thank you, Petra, for that question. So we haven't specifically analyzed GC versus AT-rich genomes, but we have done CORALL library preps on many organisms, and it works equally well for different organisms, also for bacteria. And we did never observe any GC bias. In terms of replicates, we recommend, I would say, around 3 to 4 replicates to have a really reliable outcome. What also helps for reliable outcome are spike-in controls that you might also want to consider. So we have the SERVs, the RNA spike-in controls to spike into your RNA before extraction and library prep. That will also help you to assess the quality of your data.
So how well does this feed perform on low quality fragmented RNA samples? So basically the question whether CORALL can be used for low quality and fragmented RNA samples.
Yeah, so we have shown that slide with the low input and also for FFPE. So we have analyzed this in detail, FFPE material and also heavily fragmented RNA. So CORALL performs well on those samples and you can use it. We have some minor protocol modifications for the FFPE material. So if you have that, or have a concern about your RNA quality, you can always contact support@lexogen.com and we can provide you with some adapted protocols and support you with your experiment.
All right. How long does it take to add annotations of the new species on the BlueBee platform?
Depends a little bit on the data quality that is available, but usually we can do this within a few days.
Can I start multiple runs in parallel and how many?
It is absolutely scalable. There are no limitations. You can start all the samples that you have and they are processed more or less completely in parallel.
Okay, and then we have a question which actually came from somebody who submitted it during the registration, and it was could we find alternative splicing on cancer genes from RNA derived from blood? And this question can we answer here now, or?
I think I can overtake here. The CORALL pipeline is doing a standard analysis. Doing de novo or the finding alternative splicing is not part of the pipeline, but as Andreas already mentioned, you can download the BAM files and do then your own analysis based on the BAM files, like doing a string tie analysis on that.
Thank you very much. Great, thank you, Martina and Andreas. We've reached the end of our webinar time and we will wrap up it now. Thank you everyone for attending this webinar. I hope you enjoyed it. We will follow up shortly with a link to the recording and direct answers to the questions which were left unanswered. And have a great rest of the day and goodbye.